everyone. Welcome to St. Joseph's open web-based learning platform, SavvyLearn. And also welcome to Department of Biotechnology, St. Joseph's College for Women Autonomous, Visakhapatnam. The Department of Biotechnology, that is St. Joseph College for Women Autonomous, Visakhapatnam, presents the SMOOC, that is St. Joseph's massive open online course in plant and animal biotechnology. This course is a free online modular 8-week course for life science background students which will be offered for UG level. The course Plant and Animal Biotechnology comprises 5 modules. Today in module 1, that is plant tissue culture techniques and secondary metabolites production, and the chapter 2 will be discussed. The chapter 2 is plant tissue culture media, nutrients and plant hormones, instructed by Dr. A. Veerabrahmam, HOD and Assistant Professor, Department of Biotechnology, St. Joseph's College for Women, Autonomous, Visakhapatnam, Andhra Pradesh, India.
The learning outcomes for the chapter in plant tissue culture media, nutrients and plant hormones. After completing the chapter nutrient and plant hormones, the students can able to ascertain the basic components of plant tissue culture media and screen the various nutrients including major and minor elements and evaluate the concentrations of nutrients. So this is very important for the preparation of plant tissue culture media and designing the media for various purposes in plant tissue culture. Also categorize the chemical structures of different plant hormones including auxin, cytokinin, gibberellins, abscisic acids. So those hormone combinations at different levels, different stages. For example, callus origin, callus development, multiple shoot origin, multiple shoot development, proliferation, etc. The combinations of the auxin to cytokinin ratio or also auxin to gibberellin ratio, abscisic acid. So those played a very significant role as a course outcome, and also formulate the different concentrations of the plant hormones. So this is also very significant.
The contents for the chapter are Introduction to Plant Tissue Culture Media, macro, micro nutrients and organic supplements which are required for the plant tissue culture medium preparation, plant growth regulators also called as plant hormones or phytohormones play a very important role in establishing the plant tissue culture in vitro conditions, and factors affecting the media consistency and sterilization and media preparation. So let us discuss the first introduction to plant tissue culture media. So what is plant tissue culture media? It is an in vitro technique that uses controlled conditions to grow plant cells, tissues or organs on a nutrient-rich medium. The media contains essential nutrients, plant hormones and other components to promote growth and development. See the picture where the plants grown in an artificial medium. Here the medium supports the growth and development of a single cell or a plant cell aggregate. Let us see what are the macro, micronutrients and organic supplements. Most probably NPK are acting as the basic elements required for establishing or formulating or designing the plant tissue culture medium. Those are the nitrogen, phosphorus, potassium. Let us see first the nitrogen. Nitrogen is a crucial component of proteins, nucleic acids and chlorophyll. It plays a vital role in plant growth and development. Usually the proteins and nucleic acid and as well as the chlorophyll are the three basic macromolecules required very much in plant growth and development. Second, phosphorus. Phosphorus is an essential for energy transfer, cell division and root growth. It enhances flowering and fruiting. And third, potassium. So since it is a cationic substance, potassium is involved in water regulation, photosynthesis and disease resistance, and also promotes the strong stems and improves fruit quality. Let us see the micro elements or micronutrients which are required for preparing the plant tissue culture medium: boron, manganese, zinc, copper, iron. Among these anionic nature molecules, boron. Boron is crucial for cell wall formation, pollen germination, and transport of sugars and other nutrients. Next, manganese. Manganese is an essential for enzyme activity, including those involved in photosynthetic reactions, and also nitrogen metabolism, which are actively involved in the protein synthesis and construction of the cell walls, and other enzymes molecule synthesis, and also participates in the respiration process. Manganese acting as a cofactor in all the photosynthetic reactions and also nitrogen metabolism. Next, zinc. Zinc is involved in the synthesis of auxin, a plant growth regulator, also called as a phytohormone, that regulates growth and development and is also important for enzyme activity. Next, copper. Copper is a vital component of enzymes. Most probably copper acting as a cofactor of maximum photosynthetic enzymes involved in respiration and also photosynthesis and the metabolism of nitrogen. See, the one cationic atom is involved in the other anionic metabolism, that is copper involved in the metabolism of nitrogen. So this indicates that the basic element nitrogen, which is required for the synthesis of proteins, buildup of the enzymes, and a co-relationship of the copper along with the nitrogen. And next, iron. Iron is a key component of chlorophyll, which is very much required to sustain the chlorophyll and also capturing the photons from the sunlight, which will be fixed for the energy further. And this chlorophyll pigment captures the light energy for photosynthesis and also involved in the electron transport chain. Let us see the other organic substances involved in the plant tissue culture medium preparation, including first, vitamins. Vitamins are essential organic compounds that promote growth and development. They act as cofactors in enzymatic reaction. Most of the enzymatic reactions in the macromolecular metabolic reactions that do occur in the plant cell, and these vitamins are also essential for various metabolic process in plants. Next, amino acids. Amino acids are the building blocks of the proteins. Amino acids are the and also crucial for cell division and growth. They also play a role in enzyme activity, hormone production, and rest. Let us see what are the plant growth regulators which are very much required for preparing the plant tissue culture medium. So principally auxins, cytokinins, gibberellins are required to prepare plant tissue culture medium. Auxins
promote root growth and development originally. They also play a role in cell elongation and differentiation. In cell elongation and differentiation, increase the concentration of auxins in the plant tissue culture medium always enhances the callus formation. So let us see some commonly used auxins including indole 3 acetic acid, that is IAA, and naphthalene acetic acid, and also IBA, indole butyric acid. All these three are the naturally occurring auxins which are more frequently found in the plant tissue, and most probably auxins are present in located in tips of the root and also apical tips. Of course, some concentrations were observed or identified by various scientists at the lateral bud tips. Next, cytokinins. Cytokinins always stimulate cell division and promote shoot growth. They also delay leaf senescence. Some commonly available cytokinins in the plant cell or plant tissue including zeatin and kinetin. Next, gibberellins. Gibberellins promote stem elongation and seed germination. They also increase flower size and fruit production. Commonly used gibberellins in plant tissue culture medium preparation including gibberellic acid, that is GA3. Let us see what are the factors affecting media consistency. Since some are very important factors like acting as osmotic agents, that is sucrose and mannitol. Sucrose generally acting as a primary carbon source for plant cells in plant tissue culture media preparation. It provides immediate energy for growth and development. Next, mannitol. Mannitol is also a sugar alcohol that acts as an osmotic regulator. It's a wonderful osmotic regulator for some plant species and helps maintain cell turgor pressure and prevents cell dehydration. Let us see what is the importance of the osmotic balance. Osmotic balance is crucial for optimal cell growth and development. It ensures the proper water uptake and prevents cell lysis. So that is the reason why sucrose as a carbon source and sometimes mannitol as a carbon source are frequently incorporated into the plant tissue culture medium preparation. The other factors that affect the media consistency, pH adjustment and buffering capacity. Usually the optimum pH for plant tissue culture media preparation ranging from 5.5 to 5.8, which is a slight acidic pH. So generally the pH range for optimal growth and development in vitro conditions, and some plant species may sustain even after a 6.5 pH, maybe a hardwood yielding plantations. Next, buffering solutions. Buffers play a very significant or essential role to maintain the pH of the media throughout the culture period, because any combinations of the hormones, that is auxins and cytokinins, sometimes auxin rich medium enhances the variety of acidic conditions, even highly acidic conditions where the more dehydration occurs. In such cases, a variety of buffers including MES, MOPS and HEPS, these are the very commonly used buffers in plant tissue culture medium, which ensures the dehydration of the plant tissue culture medium. Generally, the pH adjustment will be attained in plant tissue culture medium using the strong acids or bases. The strong acids and bases including hydrochloric acid or potassium hydroxide. The other factors affecting media consistency, including solidifying agent, that is agar or gel and gum. Agar is a seaweed obtained from the marine seaweed, that is a Gelidium species or Gracilaria species. Okay, agar is a natural polysaccharide derived from a seaweed. It is a common solidifying agent used in plant tissue culture media. Agar is readily available, inexpensive, and easy to use. Generally, when agar is incorporated in plant tissue culture medium preparation, the medium is known as solidified or agarized medium. Without agar, the plant tissue culture medium can also be prepared. That medium is known as suspension medium. So absence of agar is the suspension medium. With agar is the solid agarized medium. Next, gel and gum. Gel and gum is a polysaccharide produced by bacteria. Let us compare the gel and gum and agar. Agar is a polysaccharide, natural polysaccharide derived from seaweed. Here, whereas gel and gum is also polysaccharide produced by bacteria. So it is relatively a new solidifying agent used in plant tissue culture media preparation, and it is a transparent and forms a film gel. So whenever hardwood yielding plantations are to be cultivated or propagated through the callus culture or a suspension culture, frequently gel and gum is used. So which gives a film attachment of the and film support, and also ensures the consistency of the media constituents, including major and micro elements, and also as well as organic substances. It is also a good alternative to agar for some applications. Example, hardwood yielding plantations. Let us see, after preparation of the agar medium or a suspension medium, the media has to be sterilized. So by using a variety of sterilizing methodologies, that is physical methods, chemical methods, or radiations. So sterilization is very crucial for plant tissue culture medium success. Usually an autoclave comes under the physical mode of sterilization, in which the media is sterilized at 121 degrees centigrade and 15 psi pressure for 15 to 20 minutes duration. In an autoclave, the working principle is boiling point of the water rises the water vapor inside the steel jacket or simply a pressure cooker, simply a kitchen pressure cooker. The raised water vapor pressure inside the stainless steel jacket hits the surfaces of the articles placed inside an autoclave or a pressure cooker. So the surface is completely devoid of the or free from the bacterial contamination or free from the contamination or either bacteria or fungal contamination. So the mode of action of the autoclave, that is a moist heat, generally it breaks down the cellular proteins of the microbial forms, and most probably the gram negative and gram positive bacterial cell walls can easily be broken by the moist heat, that is at 121 degrees centigrade and 15 psi pressure for 15 to 20 minutes. And the other sterilization, where filter sterilization, that is where the heat labile or heat labile molecules which cannot be sterilized in an autoclave, that is a moist heat or either a chemical sterilization, can be sterilized through the filter sterilization. Heat sensitive components such as, for example, vitamins and hormones are being sterilized through the filter sterilization, in which a variety of filter is used frequently. The size of the filter is 0.02 micrometer in size, which can sterilize these vitamins and hormones. So both are the organic substances which will be required for the plant tissue culture media preparation and very much required for establishing the cultures in vitro conditions. And their aseptic techniques. So these aseptic techniques include either a physical mode of operations or a chemical mode of operations during the media preparation to avoid or prevent the microbial contamination further. Generally, aseptic technique involves the in absence of the physical modes of techniques, including alcohols, acids, or acid alcohols, and metal halides, metal compounds. Best example, lead and mercury chloride can be used as a aseptic, chemical sterilizing agents. So after complete sterilization, either way of the physical mode of sterilization or a chemical mode of sterilization or a filter sterilization, the media constituents must be optimized. The optimization aims to refine and enhance the tissue culture process to improve the plant growth, development and regeneration capacity of the inoculated cells on a defined medium. Generally, it may involve adjusting the composition of the media, the growth conditions, or the timing of the specific steps. Let us see in each in detail the media constituents. Generally, the media is uh, uh, prepared with the major and minor elements for our convenience. Murashige and Skoog, both are the scientists who designed the or formulated the both micro and macro elements into five categorized into five sectors, that is ms1, ms2, ms3, ms4, ms5. So maximum of the cationic chlorides, carbonates, halides, fluorides, bromides are incorporated into all these ms1 through ms5. And growth conditions, generally 25 degrees centigrade plus or minus 2 degree is the optimum temperature, and 5.5 is the optimum pH, and a relative humidity, for example 75 relative humidity is maintained. And if it is based on the objective, if it is a callus cultivation or a callus growth and development, uh, the growth regulators are optimized. That is, if it is a callus, increased concentration of the auxins and less concentration of the cytokinins will be incorporated into the media preparation. Of course, after sterilization of the media, either way of the physical or a chemical or a filter sterilization, the heat labile, that is a heat sensitive molecules, that is growth regulators, will be added at the room temperature. Next, troubleshooting optimization generally depends on the comprehensive understanding of plant physiology and the factors that influence the plant growth and development in vitro conditions. So by diligently monitoring, carefully monitoring the cultural conditions and responding to any deviations, one can significantly improve the success rate and efficiency of the plant tissue culture. Let us summarize the present chapter, plant tissue culture media preparation. Culture media are largely responsible for the in vitro growth and morphogenesis of plant tissues. The success of the plant tissue culture depends on the choice of nutrient media. In fact, the cells of most plant cells can be grown in culture media. Basically, the plant tissue culture media should contain the same nutrients as required by the whole plant. It may be noted that plants in nature can synthesize their own food material. However, plants growing in vitro are mainly heterotrophic, that is, they cannot synthesize their own food. That's the reason why all the macro and micro elements in addition to the organic substances in plant tissue culture media are formulated in advance based on the objective. The salts are designed, the salts are formulated. If anything goes wrong with the formulation or design, the further growth may be retarded at any stages. If it is a callus culture, of course, the first phase callus may be obtained, and then secondary subculturing, the callus may not goes further and develop into further callus. And the major types of media available in plant tissue culture medium: MS medium, that is Murashige and Skog medium; B5 medium, which was formulated by the Gamborg; and white medium, also niche medium. Among these, MS medium is generalized medium for all the plant species and can establish the plant species. And the culture media usually contains generally inorganic, organic and phytohormones. So both the inorganic and organic substances are comprising macro and micro elements that supports the growth and development. And coming to the phytohormones, those can decides the fate of the culture. It depends based on the objective taken. And the carbon source included in the media preparation is either a sucrose or a mannitol. Sucrose or a mannitol are the prime choice for the media preparation, because the sucrose has to hydrolyze by in the plant cell itself only. And of course, in mannitol also used as a carbon source for its growth and development. Agar used as a gelling agent that holds all the organic and inorganic as well as the phytohormone concentrations in intact form and make available to the plant species. I mean a plant cell or a callus cell or a meristematic cell. And sterilization will be possible with the moist heat or a dry heat mode. Moist heat, for example, an autoclave, simple autoclave or a simple kitchenware pressure cooker can be used. In dry heat mode, maximally glassware, metalware can be sterilized effectively. And these are the references for the topic plant tissue culture media preparation, and go through the references further about the topic. Thank you for your attention in the chapter.